we're going to explore the Pharma portal, which is perfect for drug development and clinical trial stratification. Once you receive your account, you can log in through our secure login page. A one-time passcode will be sent to your email, allowing you to access the portal. Your dashboard will display information about the total number of analyses you have, including those that have been processed, those in progress, and the completion status of completed analyses. We provide demo data for first-time users, allowing them to explore and experience how our system works. Let's start by uploading your data. To do this, navigate to the Services section, where we can process two different types of data, mRNA and microarray data sources. Select RNA sequencing, then click Add New Analysis. Here, you can choose the sample type, blood, tissue, or pap smear. For this dataset, we will use the pap smear. Here, we can also select the species, and finally, the group type. One of the benefits of the F420 portal is the availability of built-in controls. These can be used if the user does not have their own control set. Here we'll use custom controls. Next, we fill in the details for how we want to normalize the data and handle any missing values. If you're unsure, the default options are a good starting point. Here, we upload the dataset itself. Additionally, when using custom controls, you will need to upload the experiment design file, sometimes referred to as the metadata file. This file provides information about the groupings and ungrouped data. Currently, we can see that the data is being processed. While the data is processing, we have a few options. You can refresh the page to check if the status has changed, download the initial data, edit the name, or stop and delete the current analysis. Once the analysis status changes to complete, we can view the results. To do this, click the View Data icon, the eyeball, which will take you to the dashboard for the specific sample. Here, you can review the analysis details on the screen or generate a report in a convenient PDF format. Let's start with the analysis details. The first tab you will see is a table displaying the raw data used for the analysis. For custom controls, this refers to the data you uploaded. Now let's begin with the tabs. The first one is the Pre-Process tab. The first table we are looking at is the processed data, which represents the data after normalization. You can search the table for any specific gene of interest and also download it for further analysis if needed. The next few plots are various graphs used for quality control. The first one is a bar plot representing the raw counts across each sample. Next, we have a box plot of the normalized data to ensure that all samples are comparable. The density plot shows distribution of normalized expression. The scatter plot allows you to compare two samples. Next is the dispersion plot. Under the QC tab, there are additional tabs to help characterize the distribution of genes in your dataset, as well as the distribution across different chromosomes for both raw and normalized counts. The final tab in this section is the Individual Plot tab, which lets you compare the counts of individual genes. You can do one at a time, or you can do multiple. Let's take a look at TP53 and MYC. In the upper right, there's a legend indicating what the colors represent for each sample. You can view the normalized counts for that gene. Next is the Clustering tab. The first section simply allows you to download the clustered data. This density plot shows the distribution of the standard deviation of gene expression across samples. The heat map and tree plot in this section provide different visualizations of how the samples cluster based on their expression profiles. The correlation plots show the relationships between the samples. We can hide the legend and annotations for a clearer view, or zoom in on specific regions as needed. The plots in the PCA section offer useful tools for identifying how the samples are clustered. The first graph shows a 2D PCA and allows the user to select which components are plotted. The 3D tab displays an interactive 3D version of the PCA, letting the user rotate and zoom in as needed. The PCA tools help illustrate how much variance is explained by each component. Additionally, the MDS and TS/NE graphs provide alternative methods to the PCA for visualizing the data. Next, we can examine the identified differentially expressed genes, or DEGs. The first tab presents them in a tabular form, and you can also download the data for further analysis. The next graph displays the fold change of the genes as a heat map. Both the MA plot and scatter plot visualize the DEGs relative to other genes, coloring them based on whether they are upregulated or downregulated. Finally, the Venn diagram, upset plot, and gene plot quantify the number of upregulated and downregulated genes. In the next section, we can explore the enriched pathways identified based on the DEGs. The user can select which sample groups to compare, followed by the gene set or pathway dataset they want to investigate. The Pathways Data tab displays the resulting enriched pathways in a downloadable table. The next tab allows the user to view the enrichment plot for a specific pathway or gene set. The GSEA Multiplot tab shows the enrichment plot for multiple pathways, enabling easy comparison of pathways of interest. The GSEA Network plot visualizes the connection between pathways that meet the selected cutoff threshold as an interactive network, while the GSEA dot plot provides a clear visual representation of their enrichment relative to one another. Finally, the KEGG plot visualizes the pathway, highlighting the upregulated and downregulated genes to give better insight into their role within the pathway. In the genome section, the chromosome graph visualizes the distribution of the DEGs across chromosomes. The bicluster section helps identify groups of genes based on their expression profiles, especially in large datasets. The Data tab presents the results in tabular form. The heat map visualizes the relationship between the clustered genes and samples. The enrichment bar graph illustrates the enrichment of the top gene sets from the selected database, while the enrichment tree shows how the gene sets cluster. The network section provides various tools for analyzing how the identified DEGs interact within a network. The Network Data tab includes numerous tables offering insights into the genes in the network and their connections. The module plot shows the number of modules, groups of co-expressed genes, identified in the gene network and their relationships. The scale independence and mean connectivity graphs visualize key properties of the network. Finally, the gene network is displayed as an interactive graph, with node colors indicating the expression fold change of each gene and edge widths representing the strength of the connections between genes. All of the graphs in the analysis can be easily exported into a PDF report. To do this, the user can go back to the analysis dashboard and select the Generate Report option. From there, they can go to Generate New Report, where a form is provided for customization. The graphs of interest can be individually selected and their parameters set, allowing for high customizability. A preview button lets the user see the type of graph represented by each choice. Finally, the user can click the Generate Report button at the bottom to start the report creation process. Once generated, the report can be viewed directly in the browser or downloaded. The F420 Pharma Portal is also capable of analyzing a single sample, which is useful for identifying specific DEGs and enriched pathways in that sample. This process is as simple as uploading a file containing just that sample. This time, we'll use the built-in controls. The procedure is similar to the previous upload, but there's no need to include the experimental design file. After uploading the file and allowing it to process, we can check the analysis to confirm that the built-in controls have been added.